New genetic discovery illuminates male predominance in autism spectrum disorder. A groundbreaking scientific discovery has shed new light on the genetic foundations of autism spectrum disorder, ASD, particularly addressing the long-standing question of why the condition affects males at higher rates than females. Researchers have identified a previously unknown genetic connection through the DDX53 gene, marking a significant advancement in our understanding of autism's biological underpinnings. The breakthrough research, recently published in the American Journal of Human Genetics, represents a collaborative effort between the Hospital for Sick Children, Sick Kids, in Canada and the Istituto Giannina Gaslini in Italy. The study's findings are particularly noteworthy because they establish a clear connection between DDX53, a gene located on the X chromosome, and autism spectrum disorder. While scientists have long known that DDX53 plays a significant role in brain development and function, this is the first time researchers have definitively linked it to autism. The investigation's methodology involved an intensive clinical examination of 10 individuals diagnosed with ASD from 8 distinct families. The research team discovered that variants in the DDX53 gene were consistently inherited from mothers and present in individuals with autism. The predominance of male subjects in the study population provided crucial evidence supporting the gene's potential role in explaining the higher prevalence of autism among males. One of the collaborators of the study, Dr. Stephen Shearer, who serves as Senior Scientist in Genetics and Genome Biology and Chief of Research at Sick Kids, as well as Director of the McLaughlin Center at the University of Toronto, emphasized the significance of this discovery. The identification of DDX53 as a crucial genetic factor, particularly in male cases, provides researchers with a deeper understanding of the biological mechanisms underlying autism and offers potential improvements in diagnostic accuracy for affected individuals and their families. The research team's investigation revealed additional complexity in the genetic landscape of autism. At the same chromosomal location as DDX53, researchers identified another potentially significant gene called PTCHD1as. This discovery was highlighted by a particularly interesting case involving a mother and son who both presented with autism requiring minimal support. Their genetic analysis revealed a specific deletion affecting both the DDX53 gene and portions of PTCHD1as, suggesting a possible interplay between these genetic elements in the development of autism. The study's scope extended beyond its initial cohort through an extensive analysis of major autism research databases, including Autism Speaks MSSNG and the Simons Foundation Autism Research Initiative. This broader investigation identified an additional 26 individuals with ASD who possessed rare DDX53 variants similar to those found in the original study participants, further strengthening the connection between the gene and autism. Dr. Marcello Scala, the study's lead author and researcher in medical genetics at the Istituto Giannina Gaslini, noted the historical significance of this discovery. The DDX53 gene had previously escaped association with any neuropsychiatric condition, making its identification as an autism-linked gene particularly meaningful. This finding not only provides valuable information for future clinical genetic testing but also suggests new pathways for understanding the behavioral manifestations of autism. The research team's work extended beyond the DDX53 discovery. In a companion paper published simultaneously, they also identified 59 distinct genetic variants on the X chromosome that showed significant associations with ASD. These variants appeared in several genes previously linked to autism, including PTCHD1as, DMD, PDAC8, PCDH11X, and PCDH19. The research also identified new candidate genes potentially linked to ASD, including ASB11 and ASB9, while highlighting the FGF13 gene's role in autism with notable sex-specific differences. One of the study's most intriguing implications concerns future research methodology. The research team discovered that DDX53 lacks a functional equivalent in commonly used mouse models, which presents both challenges and opportunities for future autism research. This finding suggests that the scientific community may need to reconsider and potentially redesign their approach to studying ASD, particularly in developing new research models that better reflect human genetic variations. The international scope of this research undertaking demonstrates the power of collaborative scientific efforts. The study brought together expertise from multiple renowned clinical and research institutions across Canada, Italy, and the United States, highlighting the global nature of autism research and the importance of international cooperation in advancing our understanding of complex neurological conditions. These findings have significant implications for the future of autism diagnosis and treatment. The identification of specific genetic variants provides opportunities for more precise diagnostic tools and potentially more targeted therapeutic approaches. Understanding the role of genes like DDX53 and FGF13 in autism development could lead to innovative treatment strategies that consider sex-specific differences in the condition's manifestation. Dr. Scherer's observation that complex neurobehavioral conditions like autism can sometimes have relatively straightforward biological and genetic foundations offers hope for future treatment approaches. This understanding suggests that while autism's presentation may be complex, some cases might be addressed through targeted interventions based on specific genetic profiles. The research also underscores the importance of continuing genetic studies in autism research. As our understanding of the genetic components of ASD grows, so does our ability to develop more effective interventions and support strategies for individuals with autism and their families. The identification of DDX53 and other genetic variants represents not just a scientific breakthrough, but a stepping stone toward more personalized approaches to autism diagnosis and treatment. This comprehensive genetic research represents a significant step forward in unraveling the complex biological foundations of autism spectrum disorder. By improving our understanding of the condition's genetic underpinnings, particularly in relation to sex differences, these findings open new avenues for research, diagnosis, and potential therapeutic interventions. As we continue to unlock the mysteries of autism's genetic components, we move closer to developing more effective, personalized approaches to supporting individuals with ASD and their families.